All right. Hello. This is going to be a video guide for using Amira to segment our 3D volumes. This is going to be largely based on the Mihir's Guide to Amira document, but I'll include some more information in this about keybinds and using some neat tips and tricks so you can follow along with the document. So this is the Amira start page. The first thing we want to do is open on this top left tab our project window, and this will be where we organize all our files. This project view on the top left side is where all our files are going to be and where we'll import modules related to these files. The properties pane below this will populate after we import files and modules, and we want to modify or manipulate the properties of those files. So the first thing we want to do is import our files that we segmented from membrane and TARDIS. We want to use both of them, and I'll go into a bit more detail why after for our DRG segmentation. Note that I should mention, this is going to be specific to the DRG segmentations and segmenting a full cell volume, but it's not always necessary that you're going to need both files. It's kind of dependent on what it is you want to segment. So first thing we're going to do is click this open data, and you'll have your window pop up. My data is saved on my desktop in here's guides, and in demo files, I'm going to open demo scores.mrc. So the scores file is my output from membrane seg. That's going to have all my membranes. And here, my Amira is automated to generate this ortho slice. It might not always generate by itself. So you select this, you can right click, and if you want to remove a module, you can click this and hit remove object. So some of you, when you import, it may look like this. If you want that ortho slice to appear, select this. If it's a favorited option, it may show up here and you can click that. But per the document, you can right click this, go to display, and select ortho slice and create. That'll do the same thing, and now you can see a Z slice of that tomogram. And this is our scores file, again, that has come out of membrane. Now that we have this ortho slice selected, our properties panel down here has populated. We're on slice number 125 right now. We can go through our different slices to see different features of this tomogram. And we can see this is really, really rich in ER networks, and we seem to have a cool autophagosome-like structure here, as well as some nice microtubules running through the length of our tomogram. We also want to import that TARDIS file now. So this is going to be demo_semantic. This is the file output type from TARDIS. Now, I want to make a note that by default, TARDIS has a file type. TARDIS outputs a file type that is different to the file type of membrane. We want to run a conversion of the TARDIS output and change that to a 16-bit integer file. So that's what this demo semantic is here. That's previously explained in the membrane and TARDIS videos. So here, notice how this imported with no ortho slice. So if I want to do that, if I want to create an ortho slice, we can go through that same process. Now, one thing I want to note is why we see this ortho slice here for demo scores and how we can turn that on and off. If you look at this module, you see this blue box that is highlighted next to the words ortho slice. You can click that to turn it gray. That turns it off. This is the viewer toggle. Turn it on and off like this. So what I want to do is I want to turn this one off and generate an ortho slice for my TARDIS file. So display ortho slice. Ooh, missed it there. Display ortho slice, create. And now we can see our ortho slice, and once again, you can go through the Z and we can see the microtubules running through the length of this varicosity. Now we have our two files imported. If I want to see them both at the same time at this top here, I can change my views to have two viewers. And what you'll notice now, I have both sides have TARDIS. Our boxes here next to the words orthoslice have transformed to have a left view and right view. If I want to show TARDIS on just the right view, I can deselect the left one. So the left one is gray, right one is blue. And if I want to show the scores on the left side, I can select that. Now I can see them both at the same time. You can select one to scroll through the Z, you can use the slice number, or you can actually use the mouse wheel. So I'm using my, not the mouse wheel, I guess. That zooms in and out. Mouse wheel zooms in and out. But you can use this to scroll through the Z slices if I want to. Scroll through the Z slices of that. And there we go. So this is how we're going to import and set ourselves up for segmentation in Amira. I'm going to go back to just a one-pane viewer now and select my scores file. So I want to take my mouse and actually select my scores file. So it's highlighted green, and you can see on the left side now, the properties panel populates with all the information of the scores file. What I want to do now is launch our segmentation window. So the top where we just opened our product window, we now want to open just the one to the right, segmentation editor. Buffer for a second, and now we're loaded in. So there's three different panes that we see. In this left side, we have our segmentation editor. There is an image and label field, materials table, and selection tab. In the middle, you have a 2D viewer of what you are segmenting, and on the right side is a 3D viewer of what you will segment. The first thing you want to confirm is in the top left under your segmentation editor that you are actually segmenting the correct image. So yes, the scores file, which is our membrane output, is what we want to segment. And the label field will automatically generate, and it'll follow this format of your file name dot labels. So this is automatically generated, and that's exactly what we want. The next thing we want to do is right under it, build our materials table. So in Amira, your classes are called materials, and these are essentially what different organelle membrane classes you want to actually segment in your tomogram. For our tomograms, we have 11 different classes that we want to segment. So I am going to create 11 different materials. Now, it's important to note that for downstream analysis of your segmentations, you want to be consistent with the order in which you create or in the order in which your classes occur. And Amira is finicky in the sense that once you create your classes, you can't change their order. So I want to be consistent with my table of classes or materials. So by default, this exterior is generated. This is our background class. We never want to touch this. Inside is the first default class. I'm going to change this, and I always start with my outer mitochondrial membrane. Now I want to create a new class. So I go down to this button, click Add, and my next class is my inner mitochondrial membrane. My next class after that is always my plasma membrane, followed by my microtubules. Now I have the mitochondria, plasma membrane, and microtubules, and I can start to move into some of the more niche membrane-bound organelles and their vesicular bodies. So I'll create my ER class and my ER vesicle class. These are for vesicles contained inside ER bodies. I want to distinguish those from just freestanding vesicles. I then have my MVB or multivesicular body, as well as the vesicles inside that. Then I have my freestanding vesicles. And the last two classes are my multilamellar vesicles, MLVs. These are autophagosomes or endosomes of sorts, and the vesicles contained inside are MLVs. Now you can change these colors if you want. I have particular colors I like to associate with my segmentations. Like I always love to make my outer mitochondrial membrane this purple. I like to make the inner mitochondrial membrane this pink. I like to make my plasma membrane a sort of like sky blue shade, maybe like that. I like to make my microtubules this green. I like to make my ER this yellow. The vesicles inside ER I leave the same. The multivesicular vesicles I leave the same. The free vesicles I like to be this sort of orange. Maybe I can adjust like that. And the multilamellar vesicles and the vesicles inside them, I leave the same. So now I've fully created my materials table and I am ready to start my segmentation process. So in Amira, the great thing is that you can essentially paint in 3D. And we do that using this magic wand tool. So under materials, where it says display control, even under that, selection, we want to go over to this fourth tool right here, which is our magic wand. Now, what you'll immediately notice after selecting the magic wand is that now our tomogram has turned this bluish purple. So this is a mask that has covered our tomogram. And the way we segment is by adjusting this mask to cover only the signal and color the signal. The first thing we want to do is adjust our settings for our magic wand. So the first thing we want to select is all slices. We want to make sure when we color something, it goes through all slices, and so we're not segmenting one slice at a time. And we also want to hit same material only. That makes sure that if we color one thing and it's a volume through many slices, they're all going to be that one same material. Now that we've launched the magic wand and we set it up, it's time to properly segment. This masking scale is what is going to determine how you segment. Right now you can see we have these domain restrictors on the left side and the right side. You can also numerically adjust them, but it's not really worth the time. We need to adjust this mask and restrict the domain so that only the signal is covered. This histogram in the middle is essentially the pixel intensities of our tomogram. Our signal is going to be the pixels with the highest intensity. So we want to restrict this histogram to the highest intensity pixels. And those are going to be the 20 to 25% of the right side. So kind of this side of the tomogram right here. So what I want to do is I want to hold down with my left button on this domain restrictor, and I want to drag this to the right through the histogram. And what you'll see is as we approach this histogram and start to move through, the background classes start to open up and it starts to leave only the signal segmented. There's still some being over segmented. I think we can go a little bit further. You want to keep it pretty thin, and maybe we'll leave it at right about there. So now you can see our mask has been adjusted to capture just the signal. Now you can choose to be very lenient with your masking or very restrictive and exclusive with your masking. One thing you can notice is if you see my mouse, there are certain maybe signals you might consider here that are not. This is just how membrane actually segmented. If you want to include some of that, you can continue to move your mask back to the left, and you can see it starts to populate a little bit more. I'll leave it like this for now. How to segment now is you are essentially going to use your magic wand to select a signal and add it to your segmentation. Now, when you do this, you want to make sure you are segmenting the correct class. So in my materials table, I'm right now on the free vesicle table, on the free vesicle class. The first thing I like to start with is segmenting my plasma membrane. It gives an exterior to the cell. So I'm going to select that, and when I do, I'm going to make sure I hit 3D so I can see it appear in this 3D viewer. I can note that this exterior part here is part of my plasma membrane. So I'm going to put my crosshairs over that and select this. On our 2D viewer, we can see the purple hue of the mask cover this region of the plasma membrane. This means all of this signal has now been added or selected for segmentation. In our 3D viewer, we can also see that in 3D. And I can actually use my mouse, my left side of my mouse, to actually drag around and see what it looks like in 3D. Once I'm happy with this and want to add it to my final segmentation, I hit this button here, A, or add. You can see that when you hover over these buttons, it also gives you the keybind. So a useful keybind is hitting A. A will do the same job. I'm just going to use my mouse right now and click it, and now it's been added. When you add features to your segmentation, they will not initially populate your 3D viewer. You will have to select and deselect the 3D option, and it will show up. So we can now see this is a part of our segmentation. Let's get the other part of our plasma membrane. I'll select this side here, and now we can see the second half of our plasma membrane here. I'm going to go ahead and hit the A button to add. It doesn't appear immediately, but if I turn off the 3D viewer and turn it back on, now we have the plasma membrane of our cell. When you are segmenting, you might want to zoom in a little bit. If you use your mouse wheel here, you can scroll through the different Z's and see the different slices, but you can also now use this zoom in and zoom out feature to help you get closer on smaller things. So I might zoom in here and try and get a good segmentation of this freestanding vesicle here. I'm going to select my vesicle class, I'm going to turn it on in 3D, and I'm going to hit that. And one thing I notice is, okay, that's not the vesicle I thought it was. It actually seems to be capturing some sort of ER network. So maybe I want to lay off on that right now. To deselect your segmentation, you hit the button C. C is for clear, and it gets rid of it and deselects it. This one for here, I'm pretty sure is a vesicle, so I can click that. And what do you know? I look at that, look at it, look around, it looks like a vesicle. So I'm going to hit A and add it. I can 3D view on and off, and I see my vesicle populate. You can do the same with these vesicles here. Hit add, this one, click A for add, or just select it. Now I can see my vesicle is populating the segmentation. This is how the general segmentation process works. You can choose to be more liberal with your masking and get more signal, or you can choose to be conservative and get less signal. A kind of technique I like to use to help make segmentation as fast as possible is I will try and go to a very low masking. So the signal is very weak. I want to segment this ER right here. So I'll select my ER class, toggle 3D, and in this small region, I'll hit it. So you can see that there's this huge kind of ER piece here this ER membrane, but I'm only segmenting this purple. So we're kind of going to reverse the steps here and segment first and now adjust the masking. So I've selected that piece and I'm now going to slowly adjust the masking. And we can see both in the 2D viewer, it starts to select more signal here by turning purple, but also in our 3D viewer starts to pick up more. And I will continue to kind of move my mask further and further until I feel happy with my volume. Whoa, so now I've seen it picked up too much. I want to zoom in on my 3D viewer. So you can use either this tool on the top or you can hit the Z button. Oops, not the Z button. You can use your mouse wheel, sorry, to scroll in. Let's see which parts of this are actually ER. So that vesicle right there doesn't seem to be ER, nor does this microtubule part. So we've gone a little bit too far in our masking. Let's go back here to our masking scale and move it a little bit back. This part we're pretty sure is ER. We can adjust in smaller scales. Still ER. Too much. Maybe a little too much. I just want to focus on this part right now. If that other part under it is also ER, we can get it in another slice. So I'm pretty happy that this is an ER membrane and I can now add it to my segmentation. So I'll get on and off, and now this ER has been added to my segmentation. So this is the general process of segmenting, and the only question now is, why do I need two files? If I scroll through some of the Z, well, we should be able to see microtubules being effectively segmented through here. I can be a little bit more liberal with my masking, and I'll start to zoom in down here, and you can see that this microtubule right here, where my mouse is, has been segmented pretty well. So here we'll show why TARDIS is necessary. The segmentation that the file that we're currently segmenting is membrane, which is designed to segment membranes only. This is also previously explained in the video. If I try to segment this, notice how the microtubule seems to become a membrane. Microtubules are actually filament structures. They are not membranes. So this segmentation of a microtubule actually misrepresents what the microtubule looks like. So you'll see our microtubules don't look like those kind of membrane things. So a new thing to learn, how to get rid of things. There's two ways you can eliminate segmentations that you are not happy with. The first is whatever you just segmented, click it again. It'll highlight purple in your 2D viewer. You will not be able to see it in 3D though. And where the button that we use to add, you can actually click the button to the right of it, subtract, or hit S, and that'll get rid of it. So you can now see in the 2D viewer, it's no longer segmented. I should note that when you segment something, it has kind of the color hue of what it's been segmented as. Oops. And if I toggle on and off, that's no longer here. The other thing you can do, and this may not be as useful here, but it's more useful in close dense regions when you want to eliminate a particular class, is where the magic wand tool is in your selection. You can select the lasso tool to the left of it. You'll notice your mask disappears, but on your 3D viewer, you can actually just kind of draw a circle around the region that you want to eliminate from. And a red sort of volume will pop up. And I can click my viewer there, and I can view this kind of volume. So this entire red highlighted volume, I'm currently selected on my microtubule class, so any microtubule in this red region will be eliminated. So with this, I hit my subtract and turn off and on the 3D, and it's gone. Boom. If I go back to my magic wand tool, it's not there. So that's how to remove things and why we don't use membrane for microtubules. This is where we actually use TARDIS. So I'm going to first zoom out so we can see our full tomogram again. And the way we toggle between membrane segmentation and TARDIS segmentation is going back to the top to this image. So right now we have our scores file open. I want to now hit that toggle button and select my demo semantic file. You'll see my image has now changed to show me the segmentation of TARDIS. The plasma membrane stays, and any segmentations we previously had will stay. It'll show their outline. If I scroll through some of the Z slices, maybe we'll see that ER that we segmented. So here are the vesicles, and here's the ER that we segmented. And you'll notice that the masking has already automatically adjusted itself. So my microtubules, I want to segment using TARDIS. The 3D viewer, nothing happens. I want to return to my XY view rather than coming around, so I can hit this XY button at the top, and boom. Now, let's start segmenting our microtubules. I've hit one there, and boom, we can see this full microtubule run through the length of this cell. I can hit A or click my add button, and it's been added. So you don't always, if you're segmenting multiple features consecutively of the same class, you don't always have to toggle this on and off for the 3D. I've segmented one microtubule. When I select my next microtubule, it'll show up in the 3D viewer. So boom, that's shown up. I've selected this. Here's my new microtubule. Hit add, select this, add, like this, add, and I can continue to scroll through my Z's till I get some good representations of microtubules that I missed. So like microtubule seems good enough. Some will be fragmented. Microtubule, microtubule. See if we can get some more longer ones. So yeah, here we go. Microtubule, microtubule. And you get the gist of it. Whenever you're done, you go back to your scores file, and the mask will return here. Again, you want to change your histogram like this and go back to segmenting how you were. I'm going to kind of take a pause here and let you segment on your own, and I will come back and return with my full segmentation where we can talk about what we see in this segmentation and how to visualize stuff further in our segmentation. One other keybind I wanted to quickly share that I realized was I'm using this current mouse tool to move around in 3D. I'm using my mouse wheel to zoom in and out, but if you actually hold down on that mouse wheel, you can drag this. I can move around like this, drag to left and right, up and down like this. We'll return with our full segmentation. Okay, so hopefully you've had fun segmenting this very rich tomogram, and we're back. One thing you will note that coming back from the segmentation to the project window is you now have your demo scores.labels file attached to your scores, and this has been generated. This is our segmentation file where everything is stored. And if you can see in your properties here, you kind of get a preview of what that looks like. What I want to do is now view this in 3D. So first I'm going to turn off my ortho slice by hitting this blue, turning it off. And now you can see it's already one of my favorites. So it's up here, but we can right click this, go to display, and hit volume rendering. That is now going to generate a 3D volume of our tomogram. And you'll note that the colors are not the same as the colors in our segmentation editor. That's because the color map is different, but it's, pretty enough. So there's a lot of very interesting features of this tomogram. We can see there is a very, very rich ER network that runs through our microtubules very beautifully go through this ER network, and there's this full of vesicles. There's a very interesting ER vesicle inside ER there. And we also see this autophagosome-like structure here with the double membrane and material inside. We can't confirm if this is an autophagosome, which is why we label it as multilamellar vesicle or vesicle multilamellar the vesicles inside multilamellar vesicles. But now you have a visualization of your 3D segmentation. If you want to kind of see how your segmentation is relative to a slice, you can turn your ortho slice back on, and now you can see for any given slice how your segmentation compares. So you can see there's a little bit of signal here that we didn't exactly segment, but I can select my ortho slice and move through the volume to kind of see how my segmentation comes to life. It's really, really cool if you want to include this in any sort of presentation to kind of see some sort of visualization like this. You can even animate this rather than dragging it. If I right click on my ortho slice and go to animate, I can select this module animate ports, and that will now generate me this kind of timeline that I can use. If I just hit this play button once, oh, I need to go back to the beginning, I think. I hit play, it'll automatically run through the entire thing. If you hit this button, it'll actually go in reverse, and it'll play through the whole thing. If you want it to keep going forever, you can hit this drop down and loop it. Now, when I hit play, it'll run through all slices, and once it gets to the bottom, go over again. So the last thing we want to do after all of this is make sure we save our segmentation. So we want to select our scores.labels file, go to File up top, and Save Data As. I'm going to save it right now in my demo files, and the file name will be demo scores.labels, and this file type in Amira is going to be the .am file. I'm going to save that, and it'll be saved in my demo files. Now this .am will appear next to it, and this .am file is what I can re-import into Amira if I want to change any segmentation. For downstream analysis with the morphometrics pipeline, we also want to make sure we export this as a .mrc 2014 file. So again, I want to select this file. It says save data as. I actually want to export this data. So in the same demo files folder, I want to save this, but I want to save this file type. I'm going to select this. In my dropdown, I want to select MRC 2014. This is the type of file that will be used for our morphometric analysis to learn about the morphology of our segmentation. So now I can hit save, and now we've saved everything, and we are complete with our Amira segmentation.